Hi, my name is Jasmine Spotsville and I will be talking about the cell cycle and its internal and external control systems. Later in the video, I will also discuss how loss in control of these systems can lead to cancer. So let's get started. In order to discuss how the cell cycle is controlled, we must know a little bit about the cell cycle itself. The definition of the cell cycle is the life of the cell from the time it is first formed during the division of a parent cell into its own division into daughter cells. The process of the cell cycle consists of two main phases, interphase and mitotic phase, also known as M phase. Interphase accounts for about 90% of the cycle and consists of three subphases, G1 phase, S phase, and G2 phase. Phases G1 and G2 are mainly occupied by cell growth, and in S phase the chromosomes within the cell are copied. Mitotic phase can be broken down into two subphases, mitosis and cytokinesis. Mitosis can be broken down further into 5 stages. Starting at interphase, the cell enters prophase. Prophase is the stage at which the chromosomes are duplicated and appear as sister chromatids. Pre-metaphase, which is not shown in this diagram, is when the nuclear envelope begins to disappear. Metaphase is when the chromosomes line up on the metaphase plate and are connected to the spindles. Anaphase is when the sister chromatids are pulled apart and the cell begins to elongate. And telophase is when the daughter cells begin to form but are still connected, and the other structures of the cell begin to reappear. Cytokinesis is the final stage where the cell divides into two daughters. The various complex stages of the cell cycle causes people to wonder what regulates the cell cycle and ensures the phases are being carried out at the proper times. This question brings us back to the cell cycle control system. The cell cycle control system is defined as a cyclically operating set of molecules in the cell that triggers and coordinates key events in the cell cycle. The cell control system has checkpoints that can be regulated by internal and external signals. The three most important checkpoints are found in the G1, G2, and M phases. An example of an internal mechanism working at the M phase checkpoint is the transition from metaphase to anaphase. As long as some chromosomes are still unattached to the spindles and not lined up on the metaphase plate, the process of anaphase will not begin. Only when the chromosomes are lined will the spindles separate the sister chromatids. Two of the main regulatory molecules in the cell that are also considered internal regulators are the proteins cyclin and kinase. Many kinases used in the cell cycle are always present in the cell but must be activated by cyclin. These kinases are referred to as cyclin-dependent kinases. The synthesis of cyclin begins at late S phase and continues through G2, as seen in the diagram. When the cyclin begins attaching to CDK, MPF is produced. MPF is what promotes the start of the mitotic phase towards the end of M phase. The levels of CDK and MPF begin to drop and the cycle restarts at G1. Now to the external mechanisms. A regulator that would be considered an external mechanism would be the presence of essential nutrients. If cells are lacking nutrients, they may not be signaled to go to the next process in the cell cycle. An example of an external mechanism working at the G1 checkpoint is PDGF. PDGF stands for platelet-derived growth hormone and is required for the division of fibroblasts, a type of connective tissue. Without the signal from the fibroblasts that the PDGF is present in the cell will not be able to pass the G1 phase and divide. Lastly, I will discuss how cancer can result from disruptions in the cell cycle control system. Cancer results from the uncontrollable division of cells, which in turn forms tumors. One hypothesis that could explain how the cell is able to divide is that it does not need or produces its own growth hormone. Another hypothesis is that cancer results from an abnormal cell cycle. In these scenarios, the underlying basis of the abnormality is a change in one or more genes that alters the function of their protein products, resulting in faulty cell cycle control. If the cell cycle is not able to monitor its divisions, it will continue to divide, in turn growing a tumor. This possibility demonstrates the importance and the delicacy of the cell cycle and its controls. I hope this was helpful.